Okay, in this video tutorial we're going to look at the process of translation. So looking at translation, translation occurs in the ribosome. Now just to recap from where we left off, so in order for DNA to be expressed, a gene, a coding sequence of DNA that codes for a protein or a characteristic, must first be transcribed. We must make a temporary copy of that gene in the form of RNA, and that can leave the nucleus and then be translated. So firstly, let's look at that term, translation. So translation, as the term suggests, we're now changing the language. So we're gonna convert these sequence of bases, the A's, T's, C's, G's, into some other sequence, some other language. And we'll look at that in a second. So translation basically is a process that occurs in the ribosome. So the first thing that happens is when the messenger RNA leaves the nucleus, a ribosome, which is made out of two parts, a large subunit and a small subunit, locks around the messenger RNA. Now this ribosome itself actually contains some ribosomal sequences that I believe come from the nucleolus, that dense spot in the ribosome, in the nucleus. The ribosome itself is actually over 50%. I think it's about 55% RNA. We call that ribosomal RNA. So the temporary RNA copy that left the nucleus is called mRNA, M for messenger RNA. And then some of the RNA within the ribosome is called ribosomal RNA. And that just, there are some sequences in the ribosomal RNA that help it to interact with and find certain sequences in the messenger RNA, certain starting sequences. One in particular is AUG. So a sequence of bases, AUG, on the mRNA, is a signal for the initiation, for starting translation, that translation process. Translation also requires these special tRNA molecules. So if we look here, this is we've got a stylized picture of a messenger RNA with a ribosome that's kind of latched itself around that messenger RNA, and a tRNA sitting in here. Now the tRNAs actually look a little bit more like this, although this is a 2D version, it's kind of been flattened out onto the page. tRNA, again, it's a type of RNA. The T in this case stands for transfer RNA, and not because they're a T shape, but because they transfer amino acids. So a tRNA at the top here always carries an amino acid. An amino acid is a type of is a type of chemical. And as you hopefully know, or if you don't, watch the video on the video I've done on proteins. A protein is made up out of a chain or a sequence of amino acids. So the tRNAs here basically carry a specific amino acid. So there are I think it's 20 different amino acids, and there are different tRNAs that each carry a different amino acid at the top here. Now at the bottom, so the tRNA is actually made out of a strand of RNA that's folded back in on itself. So what's actually happened is some of the bases on this part of the strand are actually hydrogen bonding with some of the bases on this part of the strand. It's sort of folded back in on itself, and we've got hydrogen bonds here, hydrogen bonding between those nucleotides, or between the bases here. So there are actually some regions here where it's actually double-stranded, because the single strand is kind of folded back in on itself. So we've got these hydrogen bonds holding it together in this sort of T shape. And at the bottom here we'll have three bases that stick down that can hydrogen bond with the messenger RNA. So these are what transfer amino acids into the ribosome. So if I come back up here, essentially what these tRNAs do, and here it's just shown as a cylinder, is they translate. So remember, translation means changing language. A sequence of bases here into an amino acid. So AUG is not only the signal to start translation, the initiation of translation, it's also the code for methionine as an amino acid. So whenever you've got the sequence AUG on the messenger RNA, a tRNA will pop into the ribosome and transfer into the ribosome a methionine. So in this way, if we have a different sequence, if we had AAA on the next three, then a tRNA carrying a different amino acid will come in and bring in a different amino acid into the ribosome. So those are the key components in the process of translation. Now, it's worth noting that remember we call 3 bases on the messenger RNA are called a codon. So a sequence of 3 bases, and this is why it's grouped into 3, why it's important. 3 bases on the DNA was called a triplet. 3 bases on the mRNA is called a codon, and 3 bases on the tRNA are called an anticodon, because they match up. So it's the codon here that will determine which tRNA comes in with which anticodon, and therefore which amino acid is transferred into the ribosome. So let's have a look at the process in a little bit more detail. So I'll zoom in a little bit on this. Okay, so here we go. So the process of translation, basically, the messenger RNA enters the ribosome and the ribosome clamps around it. Now the messenger RNA is pulled through the ribosome, sort of like ticker tape is pulled through it, if you've ever used a ticker timer. But think of this as just like a little ribbon being pulled through the ribosome. And as it's pulled through, the tRNAs are matched up to these sites. So a ribosome actually has 3 sites. Now you don't need to know these, but we've got the attachment site, the peptide site, and the exit site. So the attachment site, or A site, is where the tRNAs come in, and basically the anticodon is matched up with the codon, the bases on the messenger RNA. And if it doesn't match, then the tRNA can't actually sort of hydrogen bond there, and it will fall out again. And so it's only when the correct tRNA, the one with the complementary sequence or complementary anticodon, comes in, that it will actually hydrogen bond and actually stay within the ribosome. So essentially what happens is a tRNA with a complementary anticodon sequence, or the complementary sequence to the one on the codon here, will enter the ribosome at the attachment site. Secondly, what happens is then the whole thing, that the mRNA gets pulled through slightly further, so this tRNA gets moved into the P site. And as that happens, essentially what happens is a peptide bond forms between the amino acids. So that this, you can see, if I just go back a little bit, just give me one sec. If we look up here, you can see we had this square amino acid here. So we've got a chain of amino acids which is starting to come out of the ribosome up here. All right. If we look down here, what happens is a peptide bond is formed between these amino acids. And so what that means is that essentially we've added the square amino acid onto the chain. So you can see now the square one's been added onto this end. And so the whole thing moves over, the tRNA in that A site moves into that P site as that peptide bond is formed. So the next step, what happens next? As I said, there we go. The whole thing moves over, coming down here. All right, the whole thing moves over, and we've now got an empty tRNA, or sometimes they're referred to as uncharged tRNAs. Now, it's a bit of a poor term because it's got nothing to do with being positive or negatively charged. It just means it's not carrying an amino acid. And this will go away to pick up the same amino acid that it was carrying before, the amino acid specific to its anticodon. So that way we always ensure that this specific sequence of bases, this codon on the mRNA, always codes for the same amino acids. So let's go through those steps one more time. So step 1, a ribosome has 3 sites. tRNA molecules enter, so tRNA enters into the A site here. All right. Number 2, a ribosome induces the formation of a peptide bond between the adjacent amino acids. So this forms a polypeptide, which will fold into a protein. So onto step 3. The ribosome then moves along the mRNA strand in order to allow the next tRNA to come in, and that's actually, you don't see that until, so imagine it's moving, and then you get to here, so these are moving across. So once they've moved across, they end up in this position. So step 4, our tRNA now sits in the P site, awaiting another tRNA to enter, and this previous tRNA here is released from the E site and is recycled. It will go pick up another amino acid. So essentially what's happening, and sometimes this helps students to kind of picture this, this is basically a process where you've got a tRNA, so I'm going to draw it as a little T shape. So it's got its little, rather than as the blue cylinders. So this is a tRNA as well. A tRNA carrying an amino acid, so I'll draw that as a blue circle. So this is the tRNA here, and this little bobble bit on the top of its head is the amino acid, and the 3 little base bits sticking down the bottom here are the anticodon. So these tRNAs come into this side, as the messenger RNA is pulled through in this direction. So this tRNA comes into the site, matches up, and its amino acid is attached onto this chain, so a peptide bond will form from the amino acid here. And then as it moves through into this position, another tRNA can come into there, and eventually it will be released on this side. So let me just clean this up a little bit. Get rid of those. So on this side, on this side here, we have the mRNA entering, the RNA, the little ribbon here being pulled through the bottom. We have the tRNAs entering with amino acids, and on this side we've got tRNAs leaving without their amino acids, and the mRNA, once it's done with, being pulled through out in this direction. So we've got messenger RNA and tRNAs with amino acids coming in this end, tRNAs without amino acids coming out this end, and the mRNA coming out that end, and the amino acids that were on the tRNAs coming in this side are coming out the top here as a chain of amino acids. So they've been linked together. And the sequence of these amino acids has been determined by the sequence of bases on the mRNA down here. So this is a fairly complex concept, and it's really hard to show with static kind of images. In the next little video clip in this playlist, if you watch this video as part of one of my playlists, I've got a really awesome animation that shows it really clearly on the inside of the ribosome. So hopefully that will help clarify it too. So an important note, actually I might come back to this point, just because I've got another picture that shows that process, so maybe that'll make it clearer. So just to kind of summarize the story so far, going into the nucleus down here, so the nucleus, the purple bit, we started with transcription. So we made a temporary copy of the DNA, which is called RNA, this temporary RNA copy. That leaves the nucleus as messenger RNA. This messenger RNA enters the ribosome. So in this diagram, it's actually being pulled through in this direction. And so what we have is charged tRNAs carrying amino acids entering in from this side, and we've got the messenger RNA entering in from this side. Only the tRNAs that match up, which have anticodons that match to the codons on the messenger RNA, are allowed to enter and actually stay within the ribosome. and they carry a specific amino acid. That amino acid gets joined on to the growing chain here, so we get a peptide bond is formed. The whole thing, the tRNAs get pulled through, the messenger RNA gets pulled through, and so eventually we get the uncharged or the empty tRNAs leaving out the back end here, and they'll go and pick up amino acids again. And some students often ask me, where do these amino acids come from? Well, from our diet. We eat proteins that are made out of amino acids and break them down, so a lot of these amino acids come from our diet. Some of them we can synthesize, we can make from scratch. So we've got the empty tRNAs coming out the side and the RNA, and that growing polypeptide chain coming out the top. So I hope that's clear. If you find that a little bit tricky to get your head around, watch. There's a really good animation after this. So have a look at that. So once this growing polypeptide chain, or this chain of amino acids, leaves the ribosome, eventually, once it's finished, we've got a long chain of amino acids, they will fold up into a protein. And so I'm only going to go over this really quickly because I've done a video on this as part of under the biological molecules topic. I've done a tutorial on protein and protein folding already, so go check that out if you haven't already. So you can see here is the sequence of amino acids, and they will coil up into an alpha helice or fold into sort of a zigzag pattern, which is called a beta sheet. So that's the secondary structure. So the sequence of amino acids determines the secondary structure, the shape into which they fold. And then the secondary structure, so you've got a sequence with maybe some beta sheet, which is kind of a zigzaggy pattern, and then maybe some alpha helix, and then maybe some more beta sheet. This time can zigzag all the way back, and then maybe some alpha helix, and you may have even more of this. And then that folds up, these fold up into a precise 3D structure, which is your tertiary structure. So these are just different levels of folding. Your chain of amino acids fold up into alpha helices or beta sheets, your secondary structure, and then they fold up further into your tertiary structure here. And the quaternary structure, you can see these different colored things, is often just when a protein is made out of more than one polypeptide. So the final protein may be made out of more than one polypeptide, or it may contain prosthetic groups, non-protein groups. For instance, hemoglobin has 4 heme groups. You can just make out that one. These grey bits. So this is actually supposed to be a picture of hemoglobin here, a protein that carries oxygen in our blood. So that's how it folds up into a protein. So I'm just going to go back to that, to where we were before. So hopefully that gives you an idea of the overall sequence. So just coming back up to the slide here, just a really important point is that so each codon, which was a sequence of 3 bases on the mRNA, codes for a single amino acid, and that's where you get these codon tables come in handy. So essentially, what you can do is if you know the sequence of bases on the mRNA, you can actually work out which amino acids they code for. Now you don't have to memorize this, but you may be expected in a test or exam to be able to use one of these. So essentially this is called a codon table, and so it's always using the mRNA codons. Now you'll know it's not based on DNA, because if you look, it doesn't have any T's in there, it's got U's. So this is based on the mRNA sequence. So AUG is always the first codon on the mRNA that's translated, and so it's often called the start codon, and it also codes for an amino acid called methionine, often abbreviated to MET. So if we have CUU, that will code for leucine, another amino acid, a slightly different amino acid. Or here, AGA codes for arginine, another type of amino acid. So the way you use one of these tables is if you're given the codon or a sequence of codons, you read them out. So the first one might be AUG. You find that here as met, or the start one. You might already know that one. Let's say the second one, just picking it randomly, is CCU. So what you do is you take the first of the three letters, you look on down the side here. So we take C, we know it's going to be in this row here. And then we take the second letter, so if it was CCU, we take this one, and then we know it's in this column here. So that already narrows it down to this box. And then for the third base, you look down this side, and so it's the CCU is the first one in this row. So it's this one here. So there we find CCU, and it tells us it codes for proline. Now you can just use the abbreviations, or they'll give you a codon table that has the full names. You don't need to memorize the names of the amino acids at this stage. So here you go. If you had, for instance, if you had the codon, maybe the next codon is GCG. So we go G here, it's in this row. GCU. So C is this column down here. So it's like kind of battleships, lining it up. So we know it's in this area here. What did I say? GCG. Is that what I said? GCG? Anyway, so if it's GCG, then we just find the one with G on the end, and we know it's alanine. And in fact, in this case, it doesn't actually even matter what the last base is, because if you look at it, all four of them actually code for the same amino acid, alanine. So that's often the case that the last base in the codon is often less important. It may not matter. It may still code for the same amino acid. And so this is, we call this a triplet code, or the reason we use a triplet code is basically because of the number of amino acids. So hold on, I'll just zoom out. Get rid of all that. So you might want to know that the AUG is the start codon. There are also a few stop codons here. UAA, UAG, and what have we got? The UGA are stop codons. These are codons that don't code for any amino acids and signal that it's the end. It's time to finish that's the end of the mRNA or the end of the coding sequence, and that protein is done. So essentially, the reason we have a triplet codon is basically because there are 64 different possible codons. So we've got 4 different letters to use. So like A, U, G, and C, 4 different possible letters. And if we have a code that's read three bases at a time, so you can have lots of different combinations, like AUG or AUC or AGC or UGC or GUA, it actually gives you 64 different possible combinations. You get 64 different possible combinations. And so if you look at in this table up here, the number of different combinations, UUU, UUC, if you go through, count those all up, you'll see that there are 64 of them. The reason that's important is if we used a code that was only 2 bases or read 2 bases at a time, then you would have much less different combinations. So actually, if we read the AUCG, if we read at 2 bases at a time, how many different combinations would we have? Well, AU, AC, AG, UA, UC, UG, CA, CU, CG. Basically, you end up with only 12 different combinations. So the issue is there are 20 amino acids. If the mRNA was read 2 bases at a time, you wouldn't be able to code for all 20 different amino acids. But using 3 bases actually gives you 64 different combinations, which means you've got more combinations than there are different amino acids. So there is some what we call redundancy. Some of these codons, some of the different codons, actually code for the same amino acids. So this is often referred to as the redundancy or the degeneracy of the genetic code. Basically means that there are more codons than there are amino acids. So it means that some of the codons will actually code for the same amino acid. Cool. So a little quick summary, since we're going back through the images again. So into the cell, starting in the nucleus, we make a temporary copy of a gene in the DNA. This temporary copy is called messenger RNA that leaves the nucleus into the cytoplasm. That's transcription. Translation, the mRNA, the messenger RNA, is pulled through the ribosome. We get tRNAs that carry or transfer, remember transfer RNA, transfer amino acids into the ribosome. Out the other side we get empty tRNAs and the messenger RNA being pulled through to the side. And the tRNAs that were carried through are added to the growing polypeptide chain. And because only those tRNAs with a complementary anti-codon sequence of bases to these codons or groups of 3 bases on the mRNA, because only tRNAs that have complementary anticodons are allowed to enter and stay within the ribosome, it means that ultimately the sequence of bases or the sequence of codons on the messenger RNA determines the sequence of amino acids. The sequence of amino acids determines how the protein folds up, and that determines the protein's function. And it's the function of that protein that will determine our characteristics. So a bit of an overall summary diagram here. This is quite complicated. A student of mine actually has done this. So in the nucleus you've got transcription here, making a temporary mRNA copy here in green from the DNA. And that's done by RNA polymerase. Remember the RNA piecing together enzyme, making this RNA messenger RNA copy. A ribosome here where it's translated, where the tRNAs are matched up with their complementary codons on the messenger RNA, and their amino acids basically forming that chain of amino acids coming out the other end, out the end of the ribosome here, and those folding up into the protein. So that's essentially the DNA determines the sequence on the RNA determines the sequence of amino acids, changing languages there, so it's called translation. The sequence of amino acids determines the shape of the protein, which determines its function, and it's the function of that protein that determines our characteristics. So that's basically it for gene expression, and it's an amazing process, and it is a little bit tricky to get your head around, but once you've kind of got a picture of it, and it may help if you draw out kind of a summary diagram like this, you'll find it a lot easier. One thing that's worth noting is so proteins that are kept like this one within the cell are translated on ribosomes that are just free floating, or we actually believe that now that they're attached to the cytoskeleton, but they're just out in the cytoplasm. Whereas there are another group of ribosomes which are found on the surface of the endoplasmic reticulum. So the endoplasmic reticulum actually connects up with the nucleus. So there's actually they're actually kind of folds or extensions of the nucleus that are in layers. Try and draw this sort of like this. And on the surface of the endoplasmic reticulum, we often find ribosomes too. So this would be the rough endoplasmic reticulum. We find ribosomes. And so some proteins are synthesized by the ribosomes. So this here is essentially protein synthesis, the ribosome synthesizing that chain of amino acids, the polypeptides. So some of these ribosomes on the surface of the endoplasmic reticulum make proteins. And in that case, the protein the protein is actually made into the lumen, the inside of the endoplasmic reticulum. So those proteins end up, or those polypeptides end up in here, inside the lumen of the endoplasmic reticulum. And they bud off the endoplasmic reticulum as vesicles. So they bud off from the endoplasmic reticulum as vesicles, and then these vesicles will join onto the Golgi apparatus, where those proteins are modified and packaged, and then they bud off as a vesicle again, and eventually, so they go through the Golgi. Golgi looks like a layer of flattened discs, much larger than this usually in the cell, but I'm running out of space a little bit. So those proteins will essentially be transported from the rough endoplasmic reticulum and vesicles into the Golgi apparatus, and then from there they'll be sent out in vesicles to the surface, where they're then exocytosed, where they're excreted. So this is what we call the excretory pathway. So proteins are either synthesized by ribosomes in the cytoplasm or by ribosomes on the rough endoplasmic reticulum. The ones that are synthesized by ribosomes on the rough endoplasmic reticulum, those proteins make their way through vesicles to the Golgi apparatus and are excreted. So we call that the excretory pathway. So just a little bit to add on the end, and that's why you find that, like our goblet cells, which contribute to our mucus, are excreting a lot of proteins and water. So you'll see a lot of these vesicles being sent through the Golgi and fusing to the surface, where they expel all the proteins that are in our mucus. Cool. So that's basically it. A little bit extra on the end there, and that's basically explains how we get, and that's what I think is really cool about this. This actually explains how we get from that code of our DNA, so that code of life, to what we actually see as a person. It's basically explains that connection, that bridge between how DNA actually gives us our characteristics. So I hope you found that helpful, and I think we've got a couple more to go for this topic, and then that'll be it.